Welcome to this lecture on cell and molecular biology. Today, we are going to explore one of the most fascinating and crucial processes in your body, the GPCR, cyclic AMP, PKA, and CREB pathway. If you have ever wondered how a tiny drop of adrenaline can make your heart pound in milliseconds, you are in the right place. We will build this from the ground up, starting with a simple story, moving into the biology, and finally uncovering the beautiful mathematics that make it all work. Let us dive in. To understand this complex biological pathway, let us start with a simple analogy. Imagine a medieval castle under siege. The guard on the wall sees the enemy, but cannot leave his post. This guard represents the GPCR on the cell surface. Instead of running, he blows a horn to alert a fast runner inside, representing our secondary messenger. The runner sprints to wake the sleeping general, which is an inactive enzyme. Finally, the general marches to the king, representing a transcription factor, who issues the royal decree to build defenses. This is exactly how a cell translates an outside message into a genetic response inside. Now that we have our castle story in mind, let us translate it into the formal language of molecular biology. The process begins at the cell membrane. The GPCR, or G protein-coupled receptor, detects external signals, like hormones. When a hormone binds, the GPCR changes shape and activates an attached G protein inside the cell. This G protein then acts like a switch, turning on an enzyme called adenylyl cyclase. This enzyme has one crucial job. It takes ATP and converts it into a secondary messenger molecule called cyclic AMP. These newly created messenger molecules then flood the interior of the cell. With the cell now flooded with cyclic AMP, the next phase of our pathway begins. These messenger molecules seek out and bind to an inactive enzyme called protein kinase A, or PKA. This binding activates PKA, preparing it for action. The active PKA then travels deep into the cell, entering the nucleus. Here, it finds a transcription factor called CREB and adds a phosphate group to it. This phosphorylation activates CREB, allowing it to bind directly to the cell's DNA. Once bound, CREB triggers the transcription of specific target genes, completing the journey from an external signal to a genetic response. Before we move on to the mathematics, let us see this entire process visually. Look at the cell membrane at the top. You can see the signal molecule binding to the GPCR, which spans the membrane. Notice how this activates the G protein, which then stimulates adenylyl cyclase. See the arrows showing ATP being converted into a swarm of cyclic AMP molecules? Following the pathway down, these molecules activate PKA. Finally, watch the active PKA enter the nucleus at the bottom, where it phosphorylates CREB, shown attaching to the DNA double helix. This diagram beautifully summarizes the biological relay race we just discussed. Having seen the biological steps, you might be wondering why the cell uses such a long, complicated relay race just to turn on a gene. The answer is signal amplification. This pathway is absolutely critical because it allows a tiny initial signal to create a massive cellular response. For example, during a terrifying moment, your adrenal glands release only a microscopic amount of adrenaline. If one adrenaline molecule only activated one protein, your fight or flight response would be far too weak to save you. Because of this multi-step cascade, a single molecule can ultimately activate millions of enzymes in milliseconds. Now that we understand why amplification matters, let us look at how we can calculate it. While biology often feels like memorization, the true power of this cascade is deeply mathematical. We use a sequential multiplication formula to find the total number of activated final products. In this equation, NF represents the total target molecules, like active CREB. Ni is the initial number of hormone molecules. The variables Ag, Ac, and Ap represent the amplification factors at each specific stage. If any of these amplification variables increase, the final cellular response grows exponentially, explaining the explosive power of the pathway. Let us put this formula to work with a concrete example. Imagine a single molecule of adrenaline binds to a liver cell. We are given that one receptor activates 100 G proteins. Each G protein activates an enzyme that produces 1,000 cyclic AMP molecules. We must divide by 4 because it takes 4 cyclic AMP molecules to activate one PKA enzyme. Finally, each PKA phosphorylates 100 CREB proteins. By multiplying these factors step by step, we see the signal explode. Our final answer shows that just one adrenaline molecule successfully turns on 2.5 million genetic switches inside the cell. We have just seen how incredibly powerful this amplification can be, which brings us to an important question. How does the cell turn this massive signal off? If cyclic AMP stayed in the cell forever, the pathway would never shut down, leading to cellular exhaustion or disease. To prevent this, the cell uses an enzyme called phosphodiesterase, or PDE, which constantly destroys cyclic AMP. The overall concentration of cyclic AMP in the cell is therefore a delicate balance between how fast adenylyl cyclase produces it and how fast PDE destroys it. To understand this balance precisely, we can model the change in cyclic AMP over time using a simple differential equation. Conceptually, the rate of change is simply the rate of production minus the rate of destruction. In this formula, the derivative on the left represents the net change in concentration over time. Rp is the constant rate at which new cyclic AMP is pumped into the cell. The term K times the concentration of cyclic AMP represents the rate of destruction by PDE. The negative sign indicates that this destruction process is actively removing molecules from the total pool. Now that we have our differential equation, let us use it to find the steady-state concentration. We are given a production rate of 5,000 molecules per second and a decay constant of 0.5 per second. In physics and chemistry, a steady state means the total amount is no longer changing, so we set the rate of change to 0. We plug in our given values for production and decay. By isolating the concentration variable, we find our final answer. The cell maintains exactly 10,000 active messenger molecules at any given moment, perfectly balancing production and destruction. Having calculated the steady state with specific numbers, let us now prove the general algebraic relationship. We start with our differential equation for the rate of change. To find the steady state condition, we establish that the derivative must equal 0. This means the speed of creation perfectly matches the speed of destruction. By moving the destruction term to the other side of the equation and dividing by the decay constant K, we isolate the steady state concentration. We have now proven that the steady state is always exactly equal to the production rate divided by the decay constant. We have covered a lot of ground today, so let us summarize the most important ideas. First, the GPCR, cyclic AMP, PKA and CREB pathway is a masterclass in biological communication, translating an external message into an internal genetic response. Second, this pathway is not just a relay race, it is an exponential amplifier, allowing a single molecule to trigger millions of changes. Finally, the cell maintains precise control over this explosive power by balancing production with constant degradation. You now understand both the biological story and the mathematical reality of how cells react to their environment. Great work!